Assalamualaikum warahmatullahi wabarakatuh. I am Nik Adlina Shazana. So in this video, my team members and I are going to explain on how to extract DNA from a sample. DNA extraction is a method to purify DNA from a sample, which in this video, the sample that we are going to use is E. coli culture. So for the first three steps, what we need are the sample E. coli culture, 100 microliter lysis buffer, 20 microliter proteinase K, micropipette, microcentrifuge tube, centrifuge, incubator, and lastly we need pulse vortex. For the first step, we need to have a 1.5 liter overnight cell culture, which is the E. coli culture. After that, we need a 1 milliliter micropipette and we will pipette the cell culture into a 1.5 milliliter microcentrifuge tube. And then, once it has been filled, this tube will be centrifuged for 1 minute at 12,000 G. Once it's done, the supernatant will be discarded completely. So, how can we differentiate between the supernatants and the pellets? Pellets are the concentrated particles that will stay at the bottom of the tube after being centrifuged. Meanwhile, the supernatant is the remaining solution that needs to be discarded completely before we proceed to the next step. Next, for the second step, what we need are the cell pellet tube and also the lysis buffer 11. 100 microliter lysis buffer is added into the cell pellet tube. This lysis buffer 11 will make the cell membrane and the nuclear envelope to disintegrate, which will cause many contaminants. After 100 microliter lysis buffer has been added into this tube, next, we need 20 microliter of proteinase K. Just now, I have mentioned that the lysis buffer 11 will cause many contaminants. Therefore, we need to have this proteinase K in order for it to digest the contaminating proteins. After these two solutions has been added, we need to resuspend the cell pellet by using a pulse vortex for 15 seconds. After the cell pellet has been resuspended, the cell pellet needs to be incubated in the water bath of 55 degrees Celsius for 15 minutes. During this process, students have to make sure that this solution is clear after being incubated. If not, extend the incubation time for 30 minutes and vortex for every 5 minutes.
Hi, I am Nur Salbila will be explaining the fourth step of this experiment. So, this is RNA and this is the sample. In this step, 20 microliter of RNA will be micropipetted into the sample. So, why we need to use RNA? So, by cleaving the phosphodiester link between the 3-phosphate group of a pyrimidine, RNA will efficiently hydrolyze RNA contaminant in DNA preparations. So, in a simple word, during plasmid and genomic DNA separation processes, RNA will be utilized to remove RNA. So now, the sample is added with RNA. What we need to do next is incubate it for 2 minutes at a room temperature. So in the next step, 500 microliter of binding buffer 11 with 96 to 100% of ethanol is added into our sample. 500 microliter into the sample and will be vortex vortexing it for 30 seconds. So in this step, the binding process will occur. The genomic DNA will bound to the silica membrane, and the nucleic acid will be washed by the ethanol solution in order to remove contaminating proteins, lipopolysaccharides, and small RNAs to increase purity while keeping the DNA bound to the silica membrane column. Next, we will transfer the whole content of our sample into a spin column. So, just put everything into the spin column. So, the function of this spin column will be explained later on by my fellow teammates, Aiman. And next, we will centrifuge our spin column at 12,000 G for 30 seconds in order to separate the cell material from the DNA. After 30 seconds, we will take out our spin column from the centrifuge, and then the supernatant, which is the liquid at the top of this spin column, will be discarded.
Hi, my name is Aiman Hakim Rizal, and today I would like to talk and present about the step 7, step 8 and step 9 of the experiment. Okay, before I start explaining about the procedure, I will now explain and describe about the spin column and the accretion tube. Imagine this is spin column and this is accretion tube. Spin column is the upper part when the accretion tube is the lower part. Okay, both of them must work together when doing the centrifuging processes. Okay, what is the function of the spin column and what is the function of the accretion tube? Spin column is when we want to check and collect the water DNA, while the accretion tube is when the water tube will be collected, so we can remove and discard them after the centrifuging processes. Okay, now let's proceed to step 7. Step 7 is when we add 500 microliter of the clean buffer into the spin column, and we centrifuge at 1200 G for the 30 seconds. Okay, after the 30 seconds, we can see the supernatant, so we must discard it. Okay, that's all about the step 7. Step 8 is when we add 500 microliter of the wash buffer and the ethanols into the spin column, and we centrifuge as usual at 1200 G for 30 seconds. After the 30 seconds, we discard the supernatant. Okay. Step 9. Step 9 is similar with the step 8, as we add the wash buffer, 500 microliter of the wash buffer and ethanols into the spin column, and we can centrifuge them as usual at 1200 G for the 30 seconds, and after the 30 seconds, we remove the supernatant. Okay, that's all about the step 7, step 8 and step 9. Okay, now I will pass to my teammates, which is Ahmad Fikri.
Thank you, Aiman Hakim. Assalamualaikum warahmatullahi wabarakatuh. My name is Ahmad Fikri bin Rizwan. Now we will go to the step 10. The empty column. This is the empty column. Is centrifuge at maximum speed for 2 minutes in centrifuge machine. This is the centrifuge machine. Why the empty column is need to centrifuge? Because there will be some ethanol that trapped in the spin column. This is the spin column. We need to get rid of excessive ethanol because excessive ethanol will damage the DNA in a long run. Step 11. The spin column is placed in a sterile 1.5 ml microcentrifuge tube. Remove the spin column carefully so it does not come into contact with the flow through. Add 50 to 200 microliter of elution buffer using micropipette which is preheated to 65 degrees Celsius to the colorimetric. The preheating of the elution buffer is essential for maximum recovery. The column is then incubated for 2 minutes at a room temperature, and after that, centrifuge at maximum speed for 1 minute to elute the isolated genomic DNA. This is the DNA that we got. Take out spin column and put it away. Because we just put the elution buffer, the DNA is already eluted into the solution here. Step 12, which is the final step, the isolated DNA is stored at negative 20 degrees Celsius. This is store place that we need to store the DNA to isolate it. Now, we are done to the DNA extraction experiment lab 1. Thank you for watching. Assalamualaikum.